Today we'll go through a little bit of a background on viruses. So Virus 101, for those of you who haven't gone to school, also you can consider it a deep dive and go into a little bit more information that some people may already know and some people may not, and it makes it much easier to interpret a lot of articles. So the South China Morning Post, it's a newspaper based in Hong Kong that I've been referencing an awful lot. I found it to be a very good source of information. Hong Kong's pretty, close to the epicenter, and they have pretty decent, reporting. Not always perfect, but much better than many other news sources I found. So if you look here, it says coronavirus's ability to mutate has been vastly underestimated, and mutations affect deadliness of strains. The most aggressive strains of the SARS-CoV-2 could generate 270 times as much viral load as the least potent type, and New York may have a deadlier strain imported from Europe compared to less deadly viruses elsewhere in the US. So there is some thought that the New York strain came from Italy, whereas the ones on the West Coast came from Asia directly. So they go into a lot of details about things, mutations, pathogenicity, right? And then they talk about how she analyzed the virus strains and saw how efficiently they could kill and infect cells. So let's go back first and let's talk about viruses in general. So first of all, we have to talk about genetic code. So the idea is that our body uses the building blocks of everything in our body are amino acids. They build proteins through which life exists. So the genetic code is like an architect's blueprint. You have to interpret it. You have to build it. It just doesn't do itself. Somebody has to be at work. So the body and cells have different structures for interpreting this, translating this actually into proteins, which are actually the bricks, which do something. I guess amino acids could more technically be the bricks. But anyways, Watson and Crick discovered a long time ago that, DNA is a double-stranded, right? And then you have nucleotide base pairs. So the thing is, these are all paired with each other. They could only be paired with one or another. There's no other option. So you only need one side in a sense. And then the second thing is, this gets translated into RNA, which then gets made into amino acids, which then gets folded into a protein. Whereas, the coronavirus is RNA, so it could skip a step in a sense. The whole making our protein process is very complicated. We still can't figure it out quite right. This is how it's done. So you have a genetic code that's just sort of linear, right? And translate into a linear sequence of amino acids. How does it become a protein? Well, it folds from this primary structure, which is more like a line, right? Into a secondary structure, which is more like a plane, right? Into a tertiary structure, into finally a quaternary structure, which is very, very complicated. We can't even figure out how to fold these things from amino acids yet, because a lot of it has to do with how the environment's set up. With different environments, it may fold differently. And once you get a protein, you're set. The reason we think that proteins sort of do everything is because they catalyze reactions. You know how we have catalysts? So enzymes are catalysts, except they're biological. And the idea is that there's a lot of different reactions that could happen in our body. But between what could happen and what does happen, there's a big gap. And so some things may happen once in, a year. But with a catalyst, they may happen once every minute. That changes it a lot, right? So they sort of direct how things go, what things happen. So let's go back now to talk about viruses. So this is a picture of a DNA virus, but it doesn't really matter inside. It could be an RNA. And then they have sort of a capsule that they're enclosed in. And that's sort of all about a virus. There's not much to them. There's a debate whether they're living or non-living. They can't reproduce by themselves. they need a host to reproduce. That is, they don't actually have the mechanism to reproduce their genetic code, actually to make anything. So they infect a host, and using their cell, they reproduce themselves. Some viruses have sort of a little bit different sort of structure on the outside. They're more capable of surviving outside of the body or outside of a cell compared to other viruses. But the basic concept is they need a host to complete their replication or reproduction cycle. So let's go back to talk about the idea of something clinical. So patterns of infection, right? So virulence, what is it exactly? Virulence is pathogenicity, the ability it can cause disease in an infected host. And we know that there's different amounts of virulence for different organisms. Bacteria have virulence. It's not a term specific to a virus. But the idea is that, it's not quite so simple determining virulence. First of all, measuring it is rather simple, right? So they, take some cells, right? And then so you say, okay, let's infect the cells and see how many die. Wonderful. And you compare one versus another. If you don't know this term, LD is lethal dose of 50%. ID is infectious dose of 50%. So causing 50% of the people exposed to be contaminated. Lethal is obviously death. But the idea is that you have actually here, you have different strains, right? To see how much of the cell you kill with one strain versus another strain. And that's actually what the researcher did in the article. We'll go back to that. But the important thing to realize is that virulence pathogenicity doesn't exist without knowledge of a host. It depends. It's different for different hosts, right? We all know that if you're trying to affect a mouse versus a monkey, they're going to be different. Some might be great at infecting mice, and some viruses might be horrible at infecting mice, but wonderful at infecting monkeys. It depends on the host. Just like how R0 infectivity rate and distinct measures are also related. Nothing exists in a vacuum. I think that's something that's becoming more and more obvious through time, that everything's sort of related. So if we go on to the next sort of picture, this is about the idea of even in the host, there's different areas that can be infected, right? So the idea is that, some things infect the brain. Meningitis, right? Brain infection. Some things infect the heart. It's relatively uncommon, but a lot of times we call it viral myocarditis, and some viruses are more well known to do that than others. You could actually take a biopsy of the heart and see the virus inside. A lot of stuff that infects the gut, right? Some infect the kidney. There's different parts of the body that can be infected. And then the thing is, some viruses can only infect one part, like encephalitis, meningitis. Some can infect other parts of the body as well. The coronavirus is known to attack the lungs, but there's findings that it may be other parts of the body. How exactly does that work? I think we don't know at this point. But the idea that it could only inhabit certain parts makes sense, right? It's like you have a plant that could grow in a desert. It's probably not going to do well in the tropics because there's too much rain. It's adapted to the desert. So these viruses could be adapted, but that could also change. And that's the complicated thing, right? So there's this thing called passage. And the idea is that, remember, viruses don't reproduce by themselves. You can't just feed them and say, oh, here, you want some food? Let me give you some fish food. Okay, good. You'll take care of yourself for a little bit. You have to keep them alive, keep them reproducing, and the way they do it is passage. So they could passage through animals, and they could passage through cells, just cell lines. And so they know that it changes when they go through different hosts. So for example, if we're going through animals, right, you give them to a new animal, like yellow fever. Well, in monkeys, it causes hepatitis, liver inflammation, and the monkeys die. But then they give it in the brain of mice and attacks the brain and then likes it there, is happy, it'll reproduce itself. But then you give it back to the monkey and it can't actually do that anymore. It's changed. It's adapted. Passage in cell also changes it. And so that's one issue when you go through these sort of artificial constructs, right? The idea is that we're giving an artificial environment. This is not quite the same as it's living in the wild. Wild type is a virus we just grab from people. But when you're testing in the lab, the virus may change, right? And you need enough of the virus because you need to grow it so we have enough to test on, right? But if it changes, then you may not actually be testing on a virus that you actually procured from the wild just a few days ago. It may be different by now and your conclusions are not actually relevant. For example, the polio virus, right? So, they got some from humans and they started testing them, right? And then they did the intracerebral passage in monkeys. And then they said, okay, well, we got some that are pretty good, right? So, they're not really going to be infectious when you just give it to somebody, in their mouth. So we got to try this nasal spray, right? Because this is how they're infectious. But this is the cerebral passage in monkeys. It's not the same virus that was present in humans. So the nasal sprays didn't work. And then they realized later that it's actually the oral route that's the important one, even though for this particular strain of virus in the lab, it wasn't. That particular strain of virus is almost irrelevant because it's not the real one we worry about. HIV is actually another great example, HIV-1. So in the lab, you just try to grow these things, right? You got to make sure they stay alive. So they tried it. We got it working on this one particular T cell line. And they tested it like, oh, wonderful, wonderful. So they took the blood samples from people who've had HIV and their antibodies neutralized the virus. We're going to get a vaccine. Well, you know how that story ended, right? Obviously, we don't have a vaccine for HIV, and people wonder if we ever will. But, they realized, oh, it's an error. Apparently, we used the wrong T cell line to grow the virus. We should use another type of T cell line. Because actually in the body, they don't use this T cell line, and it mutates the virus. It's very complicated, right? That's why it's good we have researchers doing this. But the thing is, we should understand a little bit about what they do. Because, sometimes they make mistakes just like everybody else, right? We know that it's sort of obvious now the CDC made some mistakes in coming up with, a test for the virus. And that's why the US was so behind in testing. Okay, so let's look at sidebar genetic determinants of virus virulence and attenuation. So the idea is that things mutate. You have these little amino acids. They're very easy to get wrong. You make a mistake, you copy wrong, right? Just like anything else, right? Viruses aren't as good as human cells in the fact that they don't have as much error checking mechanisms. So the idea is that in the human body, a lot of our cells may be mutated, but the idea is the body will say, okay, you're mutated, you're done, right? And then they tell the cell to kill itself, and it does. But the viruses don't have that kind of mechanisms, and sometimes it's good, sometimes it's bad, but they have an awful lot of mutations. Mutations are almost always bad. But in terms of the virus, they sort of don't care if one or another survives. And there's so many viruses, right? We're talking about sometimes they can make, 1 million, right, a day more. So it doesn't really matter about a few that die here and there. But the idea is that just one little change. Remember how we looked at the picture of the DNA? This thing? One little change here could change a lot. And the thing is, for those who know a little bit more about, genetic material, there's non-coding regions and there's coding regions, right? The non-coding regions don't translate into a protein. They're still useful. We're not clear exactly why. But the idea is that sometimes even in the non-coding region, one change makes it so the virus has increased virulence, right? So they looked at this for the 1918 flu, right? The one that killed an estimated 50 million people. It's not quite so simple, right? So you have this, right? And they sort of change a little bit, right? To say, let's change it from a more recent influenza, right? So you see just one little change, right? Boom. Mortality from 100 to 0. Okay. a few changes here. Huh. Mortality goes up again to 100. Okay, more changes here. Wait, mortality drops again to 0? It's really complicated, right? Could be one gene, could be multiple genes. We're not sure exactly, right? And the problem is this is still, there's no way to go back in time and test 1918. But the idea is these are only in cells, right? And a cell is just one part of the body. There are so many cells, they work together. There's all these other things going on, like these cytokines, these hormones. It's quite complicated to determine virulence, right? So practically, they could test it in a lab, but that also doesn't necessarily translate into how virulent it is in a person, right? And then the animal they think is slightly better than a person for testing, shall we say, because we don't believe in testing on humans nowadays in these cases. Except I find it interesting because we still do in certain other cases. So the idea is that if you look back, right, we have to talk about something else. They enter the host, they stay, and do they leave? Do viruses persist? Do they outstay their welcome, right? And so the idea is that, yes, some viruses do persist. There's a high titer, acute disappears, persistent stays there, latent disappears, or shall we think, and it shoots back up. This is like your, herpes, your shingles, smoldering infection, you have some low level, right? So the idea is that our body, right, has an immune response. It tries to kill all these cells. How can the virus survive? Well, either it could be immune tolerant. That means somehow the body ignores it. It could also overwhelm the body system. The body keeps fighting it, but there's just too many of the viruses to keep on clearing. And then there's the idea of latency. So when you see here we have a latent infection, it means we think it's disappeared, but it's hiding out somewhere. So, the most well-known ones are the sort of herpes viruses, right? So these are all sort of related. So the idea is they integrate into your host genome. They hide out, we say, in your brain, your neurons, your spinal cord. And so they don't have to do anything because the cell will reproduce them because they're already integrated into your genome, or they're just hiding out in the cytosol, which is outside of the nucleus. And when the body replicates a cell, they'll just replicate it along because they replicate everything inside. But it's thought that an RNA virus can't do this because you have to have DNA. We only integrate DNA because we have DNA as our genetic code. So let's look back. So, can this be persistent? Let's see. Okay, high titer replication. We have that, but we've seen that it's been killed off. Okay, smoldering infection. No. We're testing for it, and in China they did test 4 different areas, including the blood. We think it's gone. This is irrelevant, but this is talking about counting cancer. As far as we know, you can't have a latent infection with RNA virus, so we're good, right? But then you ask the question, wait a minute. If you can't have it, these people that have it again, is it reactivation or they get the infection again? So they're saying they're not immune or they still have had it? The problem is we don't know what's more right, what is absolute. Nothing's absolute. Maybe it's actually latent and it's just the virus has figured out a way to be latent. Or maybe they're not immune. I think it's pretty important to figure out which one is the truth first before we go on the other path. So let's go back quickly and review this article now. So, we'll be able to understand it more. So she says that she actually tested the virus from some patients, and they actually said that they have many mutations and they changes pathogenicity. Okay, remember we understand pathogenicity, how it can affect things. So she just took 11 patients, right? And then, she said that, they tested it and see how well they could efficiently kill and affect cells, right? So here's all the sort of errors you could do with doing a cell passage. And then they compared it to the strains in Europe, the strains in the US, right? And then she also made a caveat saying, look, looking at the clinical outcomes, the weaker strain didn't mean that everybody had it, right? Because some patients had the quote weaker strain and they got very ill. So we know that has to do with the host as well, and people have different genetics, different immune systems, right? And then so she says you have to take this in account, right? Because, defining mutations in a region, right? Well, it depends, right? Because maybe you use the vaccine for this place and it's actually developed from, a virus that's more prevalent in another place. So she said detected mutations. Remember we said mutations are just sort of changes in the DNA or RNA. In the coronavirus cases, in the RNA of a nucleotide, making it so we make a different protein. Or even if we don't make a protein, somehow it changes things. So the most aggressive strains could generate 207 times as much virus. This is one way you could overwhelm the body's immune system and continue surviving. They also kill the cells the fastest, right? So she's saying that true diversity of strains is largely unappreciated, right? And this idea is that we think what things we have are what we have in the wild. But the idea is that, wow, there may be some strains that are easier to grow in a lab than others, but doesn't mean those are the actual ones known in the wild. And here, actually, she looked at ultra deep sequencing. So the idea is like, how when you have something and you have a brief introduction and then you have a deep dive, you go into much more detail. So she read it a lot more times because we have errors in our measurement as well. And so this is the thought that, people could have missed stuff, right? And then they found sort of consecutive changes. And we know viruses mutate a lot, right? Investigating the functional impact of this trinucleotide mutation would be highly interested. And I think that's the idea, right? Yeah, how does this affect things clinically, right? It's very interesting, right? Remember how we said the 1918 influenza had lots of mutations, right? We saw just a few mutations could create 100% mortality rate. It has a lot more than just a few, right? So the idea is maybe some things are so highly mutated, they're very good at affecting other people and very good at killing cells inside the body. So this talk ran a little bit long, but this is us sort of going back into the basics of virology, talking about how viruses get tested, how research is done on them in lab, how they reproduce. And, it's important to know this stuff so we can understand the further research coming out about the novel coronavirus 2019.